construct call complex score matrix for protein complex prediction from APMS data. So my main affiliation is Institute for Infocom Research, A-Star, Singapore. I'm also holding a joint position in Nanyang Technological University, joint work with Zhipeng, Chigang, and Mingwu. Zhipeng is from Fudan University, China. Some people don't know the A-Star Singapore, so give you some background on A-Star. So A-Star is my company, is an agency for science, technology, and research Singapore. So we have two research council. One is a biomedical research council, one is an engineering research council. So each research council, we have 7 national institutes. We have quite a number of world famous biologists or scientists. For example, this Xini actually won the Nobel Prize, and Sir David Lin, I think he is a co-founder of the cancer gene P53, and the other scientists I think is quite famous. So in my today's talk, here is the outlines. So give you some background and introductions because of the title. We'll have the protein complex and the APMS data. So the concept, immediate question is what are protein complex, what is the APMS technique, and how to computationally detect protein complex. Protein complex typically consists of 2 or more proteins. Okay, they collaborate with each other to perform some biological functions. This is just give you some example: DNA transcription, mRNA translation, signal transduction, and so on. Here is one example of a protein complex, RNA polymerase II complex. Basically, it's transcribe genetic information into messengers for ribosome to produce protein. So for many computational methods, we only to do some prediction of protein complex, just identify those members in the protein complex. We're not going to predict the structure of the complex. Second question, what is APMS technique? So affinity purification, AP, is a technique to study protein-protein interaction as well as protein complex. So basically you have a batch immobilized by affinity tag, then it can pull down the price by this batch. Two methods you can use called immunoprecipitation and tandem affinity purification. After you do this AP, you need basically to identify what are these price. So basically you use this mass spectrometry, make use of the properties of mass to charge ratio to identify the price. There are 2 large-scale APMS data released in 2006. One is Gavin's group, one is Krogan's group. Okay. Give you a sense of APMS data. So this is purification name, what kind of method they are used, and given this particular batch, you get various price. So this is a same batch with all these price. Of course, they have thousands of these purification data. In this work, we detect the protein complex from the APMS data. Why? The good thing of APMS data, so basically they provide some kind of call complex membership. So multiple protein, you know the batch, they basically get the various price. It could be physical interaction, could be some indirect interaction. So but basically they tell you the kind of call complex membership. So it's more useful than those pairwise protein interactions. However, it may not be perfect to directly detect the protein complex. This is because of 2 reasons. First of all, a single batch, maybe you get various price. They may actually not occur in one complex, okay? So maybe a single batch, you get the price belong to a different complex. Secondly, as we all know, this purification data, they have large number of false positive and false negative. Some of things you detect may be wrong. Some of you do not detect. So because of these 2 reasons, we use them, but we try to detect more accurately. Computational method detect the protein complex. Last year we write a survey paper, basically is to include all those recent papers on protein complex detection from protein interaction network. Basically, they try to identify those dense regions in protein interaction network. It tends to be a protein complex, some representative method in this domain. Some research detect complex from APMS data. They basically convert APMS data to PPI interaction network, then use existing method to detect protein complex. In particular, many methods actually consist of these 3 steps. First step, I think, is compute a protein affinity score using a social affinity. This is one representative work using a purification enrichment and a dice coefficient. I will not go to the details, but basically can compute the affinity score between a pair of protein. Once you get these pairs of protein, many proteins, you give them score. Basically, because of protein-protein interaction, may not be reliable. So one more step here is try to identify those reliable protein-protein interactions. Typically, you give a score, you can rank those protein pair. However, you need to somehow to cut this. A lot of methods actually use some benchmark protein complex. Assume any pair of proteins in a complex, they have the interaction. Then you have your score ranked by the affinity score. You also have a benchmark complex. You try to match them. You get the best match. Then basically to get a threshold to cut the protein interaction to get a reliable protein interaction. Eventually, in a third step, you just use some existing method like MCL to detect protein complex. Okay, so basically we summarize some of the existing work. We think that some of this work actually more or less rely on some supervised information of reference, which is you already know some protein complex data, especially for APMS data site. So may not be applicable to other species. So basically this method is an unsupervised method, not rely on the existing protein the complex. The second issue is some of the methods they basically discard the useful quantity information. Of 2 proteins, they are unlikely to co-complex. They seldom co-occurrence in the APMS data. So basically we use all different kind of information to tell us maybe the 2 protein pair is likely to be co-complex or they unlikely to be a co-complex. Once we get this score, we can assemble this protein into complex. Our method talk about the framework and how to construct a matrix. We also have a matrix merging because our APMS data could from Gavin's data, could from Krogan's data. We try to merge them together, then we do hierarchical clustering. The framework, so basically, as I said before, this is Gavin's data. This is Krogan's data, but according to method, you can also divide it into 2 subsets. But for each data, we basically construct complex score matrix. Then we merge them together into one unified matrix, then to do the hierarchical clustering. Okay, so this is an important part of this research work. So how to construct a C2S score matrix. We have a set of purification data here. We regard each purification experiment as a piece of evidence of a co-complex relationship among protein. In particular, here we consider 4 different types of relationship. Type 1 basically is a type 1 and type 2 are bait-prey relationship, but one is positive, the other is negative. So this one basically tells us given 2 protein pair, X and Y, X is prey, Y is bait. X is bait, Y is prey, or Y is bait, X is prey. Basically tells me, okay, these 2 proteins, maybe they should belong to the same protein complex. Of course, you cannot just based on one particular purification, you need to do some statistic. The second one is on the contrary, so basically you have X as bait, but you do not get the prey, Y as a prey, or Y as a bait, X is not prey. So these 2 are bait-prey relationship, one positive, one negative. The type 3 and the type 4 is basically prey-prey relationship. 3 is positive, type 3, type 4 is negative. This means I have another protein as a bite and XY as a prey. So So maybe also tells me XY could belong to the same complex. So idea is quite simple. So across all the purification data, we basically, in principle, the more co-occurrence of these two proteins, your positive beta-pre relationship and the positive pre-pre relationship, then more likely it should belong to the same complex. Otherwise, if you find many cases, they are negative beta-pre and negative pre-pre relationship, they are unlikely to be co-complex. This is the whole idea of this paper. We have how to compute this score. For any given two pairs of protein, we compute your C2S score. This is basically log likelihood ratio. This numerator basically is a conditional property. Given the XY are co-complex, what is the probability they occur in this E? E is all set of all purification. And also we compare with random case. Okay, so this turns out becomes because the log and the probability, the term together, so is a sum. So parallel I actually represent the evidence of each of these, one of these four types contribute to CS score. Basically, you have type 1 and type 3, which is positive, then your score will be bigger. Okay, if you have a lot of negative 1, type 2 and type 4, then you become smaller and negative. Okay, negative score could be positive, could be negative. The count basically is a number of purification. You count this type of the purification, these types of evidence. Okay, so in order to compute the parallel I, we need to compute the four probabilistic parametric. Rho RP basically is if what is the probability given this is X as a bait or a prey, what is the probability Y will be detected as a prey? Okay, so this all these four probability parameters will learn from the purification data. So rho BP basically is random case for non-specific region, but maybe due to the experiment you get this false positive. Okay, so we compute this probability. This is a bait-prey relationship. Now it's prey-prey relationship. You have other protein as a bait. Given X already detect what is the probability Y as also detect as a prey, and as well as a prey-prey relationship. This is a negative one. For this is random one. For non-specific region, they also get it. So I will not go to the detail how to compute these four numbers, four probability. In our paper, we have a half page to elaborate this. But the idea here is this probability actually will be bigger. Okay, if you're really within the same complex, this one will be detected with a high probability. This one also will be bigger. Another probability after we compute will be very small. Okay, once you get these four probability, then you can compute the parallel R quite easily. This is a positive beta-prey relationship. This is actually a negative beta-prey relationship. This is a positive prey-prey relationship. This is a negative prey-prey relationship. So sum them together, actually eventually you can see what is a positive overall, what is negative overall, indicate if these two proteins should be co-complex or unlikely to be co-complex. The count actually is quite simple. The type 1 positive beta-prey relationship is a type 1. So P3, P4, actually here we can see the P4 is a prey, but P3 is not in, P4 is a bait, P3 is not a prey here, or P3 is bait, P4 is not a prey here. So basically this tell me I don't have a positive evidence to support me for the type 1. Type 2, actually, I have a lot of negative support. You can see E4, E8, these two purification data. I have this P4, but I don't have a P3. I have P3, I don't have a P4. So this is positive and negative beta-prey relationship. Here is a positive prey-prey, negative prey-prey relationship. I have one evidence tells me P3, P4, they are prey, all the preys of P1. But I also have other evidence tell me, okay, here I have P3, I don't have a P4. Here I have a P3, don't have a P4. Here I have a P4, don't have a P3. So basically, we count this, tells me how frequent these things occur. Okay, So basically, just now I just elaborate what is the process to compute any pairs of protein. What is the likelihood? Basically, they should call complex and unlikely call complex. Once we get a matrix, if you just use one data, then you get one matrix. If you use multiple data, you get multiple contracts. So basically, we weighted sum them together. In this work, basically, we treat them quite equally. So Gavin, I give the weight 1. The Krogan, you have 2, so I give 0.5, 0.5. Suppose this is also 1. Then I do hierarchical clustering, quite standard method. Here, in the beginning, all the protein, I just give them, each protein is an individual cluster. Then I compute the similarity between them. Then this method will automatically stop this iterative process. If the similarity is larger than 0, which means I should continue to merge them because I still got a positive evidence. Once you become negative, you automatically stop. So during this iterative process, I basically find this most similar cluster, merge them together, update the cluster, also update the similarity between this new merged cluster to all other clusters. Then I iteratively this process until the similarity less than 0, I stop. All the cluster will be correspond to a protein complex. Okay. Talk a little bit on the data evaluation metric and also some results on Gavin's data and the combined data. Briefly talk about this. The data set here just summarize the TAP data we are using in this experiment. The purification, Gavin's data, Krogan's data, but we also use some threshold to get a Krogan's high confidence data set. Then we combine them together. And in each of these Gavin, for example, Gavin's data, I have about 2,000 bait. This is the number of prey, and also for the Krogan data similarly. For benchmark protein complex data, I use two. One is a CYC 2008, one is a MIPS. These are all manually curated benchmark complex data for East. Here we use East data. Yeah. Some of evaluation metric, given set of protein complex you predict, as well as you have a benchmark protein complex data. So basically you try to evaluate performance by compare with them. Some of the measures defined by other researchers include some sensitivity, what is a positive predictive value, and accuracy. So we use them together. Basically, this accuracy combines the sensitivity with this positive predictive value. Idea here is just you use some predict complex. You see what is the overlap between your predict complex and the benchmark complex. You try to find some maximal match and so on. Then another one is a recall score here. Basically, is trying to see this is your predict complex and this is a benchmark complex, predict complex, compute the overlap. If this score is larger than or equal to omega, then we think they are match. Okay, this is I think in Baker's M-code method actually use this. Using the Gavin's data alone, it seems our method actually is quite good in terms of this recall. Here is omega value change, because omega value basically tells us what is predict complex and the benchmark complex are match. If omega is bigger, then which is a very strict requirement. So basically here is a recall when the omega is bigger, then you actually recall will be low. Okay, because it's more strict to decide if a predict complex match with a benchmark complex. Since our recall score is quite high. Here is comparison with this CYC 2008, the catalog, computer sensitivity, PPV, and accuracy. This red color is a C2S score. It performed quite well. Some the Gavin score actually, if you compute this, PPV is high, but the sensitivity is low. Overall, the C2S score performed not bad. And also remember, some of these methods they are supervised methods. This is use a MIPS complex as a benchmark data. We do the similar things. Since the protein complex they work with each other, they're likely to be the protein share a similar localization information as well as they perform some common biological functions. So we also compute what is a score. Basically every score they share the location and the biological process, molecular function use a GO. So seems this score also higher than the other existing method. This is some combined Krogan and Gavin score. We actually also get a good recall across different omega value. And also use this CUIC to compute the sensitivity, PPV accuracy. Okay. Let me just summarize our research. Here is basically we predict the protein complex from APMS data. We propose a novel method called C2S Score. The idea here is we not just to say if the two proteins should belong to same complex. Okay, they also sometimes shouldn't belong to same complex. This is because in APMS data, sometimes I just find one, I do not find the other one. So as a bait prey or prey. So basically it tells me what if these two proteins should be together and should not be together. Then we actually estimate some probability parameters to compute this matrix. Based on this matrix, merge multiple matrices together and perform the hierarchical clustering. Results, so it's not too bad. So some of the future work on this research we want to explore. One thing is we just get some average measurement across all the purification. However, each different purification, they should have some kind of characteristics. Maybe we should take this into consideration.
I didn't start. I think you're over time.
Oh, okay. So just say a bit more. So basically, integration, you can actually integrate in a different level, raw data level, or after you do the complex, you can merge them together also. Is another direction. And currently, because you do the hierarchical, each protein you just occur in one particular complex. Like another method, we can have a post-processing method to allow one protein can occur in multiple complexes. We are also developed a free software package, including 3 recently or paper published in this, including C2S and GSAB paper and our previous 2009 BMC bioinformatics paper. We want to make this public available for other researchers for comparison or to do some further research. Another application, recently we are doing some gene phenotype association analysis. We build some kind of protein complex network. We use some propagation algorithm. So this pretty complex could be used for this application. Thank you very much for your attention.